Sanchita Bhattacharya. I'm a bioinformatics project leader in Baker Institute of Computational Health Sciences at UCSF. Today I'll be focusing on leveraging the publicly accessible clinical trials data, basically looking at the some of the sharing efforts, dissemination, and repurposing. So the motivation for the initiatives that we are kind of working on under the umbrella of ImmPort, which I'm going to talk a little bit later, is that the clinical trials generate vast amounts of data, and large portion of this data is never published or made available to other researchers. And in recent years, there are several examples which shows that data sharing could advance the scientific discovery and improve the clinical care by maximizing the knowledge which is gained from data which are collected in trials, stimulating new ideas, and also avoiding some of the unnecessary duplicative trials. So here is a kind of an illustration of the clinical trial life cycle. And during this life cycle, there are data generated in the form of like metadata. It's the data about data and on individual participant data, summarized data. And then also during the publication, you share the data packages, and after the drug is approved, then or during the approval process, you submit the data to the regulatory agencies. So as you can see, that there's a lot of data which is getting generated, and the community, if it gets shared, then the community gets benefited. So along those lines, in the Institute of Medicine US came up with a report called Sharing Clinical Trials Data, Maximizing Benefits and Risk back in 2015. And so I'll just summarize the report. And the benefits of data sharing is, confirming the published results, generate better evidence, facilitate the additional findings, prevent duplicative trials, and ultimately improve the public health. On the other side, there are risks of data sharing, and so the privacy of the clinical trial participants is very important. Then the legitimate economic interest of the sponsors, invalid secondary analysis, appropriate recognition of the researchers for their intellectual contributions, and there's also a fear of research institutions that data sharing will be unfunded mandates. So what is important, and the point I want to make, is that we should be very careful when we share the clinical trials data, and it should be a responsible sharing. And so market and regulatory forces are driving these initiatives to publicly share the patient-level data from clinical trials, and here are other entities like pharmaceutical companies, government health agencies, and nonprofit organizations. On the other hand, there are also electronic portals which are getting built to share this kind of data for either requesting or sharing and analyzing clinical trials data. And just wanted to give you an example of clinicaltrials.gov, which most of you might be familiar with. And it's a registry, and it shares the summarized data from the clinical trials from either private or public agencies or the pharmaceutical companies and others. So there are about, today I was this morning searching over 300,000 clinical trials registered, and across 209 countries, and all this data is, at least when you run a clinical trial, you register at clinicaltrial.gov, and you get some summarized data. So, and then on the other side, I also have Vivly. Vivly is a platform through which you can get the open access to clinical trial data. It's like an Amazon marketplace. You go through Vivly and look for clinical trials which are run by either pharmaceutical, government, or nonprofit organizations, and you request the data, and then you write a proposal, and And then there's a review board. They go through the proposal, and if they agree to it, then they will share the data with you. So basically what I'm getting to is like, sharing the data increases the transparency in the field of clinical research. And on the other side, so this was all about clinical trials, and within clinical trials or on the clinical research space, we had been, and I come from that space where we had been generating lots of data in the field of immunology. And so, as you can see here, either from the humans or from the mouse model or other species, we are generating data in the field of proteomics, transcriptomics, genomics, and epigenomics at the population level, in the single cell level. These are all the high throughput technologies we use for measuring for the immune measurements. And this comprises of like cytokine measurements, immune cell subsets, and then the immune-related genes. And then, as you may know, that human phenotype depends on the age, race, gender, genetic background, and from a single person, a single participant, you can get all this data. So I'm kind of more referring to the systems immunology, where you look at the genetic predispositions, the immune repertoire using the sequencing, gene expression, and then, there are other techniques like ELISA, CyTOF for measuring the immune cell subsets, cytokines, and lately a lot you have been hearing, I'm sure, about microbiome. So from a single participant, from a single subject, you kind of get all this information. So definitely there's a data deluge. So today, I'll be focusing on one of these repositories and called ImmPort, and I had been the science program lead for this ImmPort project, which is funded by National Institute of Allergy and Infectious Diseases under the NIH umbrella. And so ImmPort came into existence about 12 years ago, and this is primarily was designed, this database or the portal was designed for NIAID researchers, those who are getting funded by NIAID, and they were required to submit the data from all the experiments they run. They generate the data, and that goes to ImmPort. And ImmPort supports the FAIR principles, which is like findability, accessibility, interoperability, and reusable. So next few slides, I'm going to talk a little bit, spend a few minutes on ImmPort. So where is this data coming from? So ImmPort redistributes the data from the major NIAID-funded programs. We have about 30+ programs, those who are submitting the data, and these are the big consortiums, those who are sharing the data, like Human Immunology Project Consortium, and then we have AMP, which is Accelerating Medicine Partnerships for Rheumatoid Arthritis and Lupus. So we are getting a lot of data for autoimmune diseases, cancer, and then much of dynes for the preterm birth research, and the list goes on. Now I'll talk about a little bit on the data flow. So we have data providers, and I just mentioned about the data providers who are the NIAID investigators. And so once they submit the data, and then the data goes to a private space, and then once they are ready to publish the papers, they distribute the data. So from all this data collected on the studies go to the shared data space, and then it's disseminated to the research community and citizen scientists, data enthusiasts. Those are the ones we cater to. So this is just a workflow. And then we also have some of these data analysis tools that we develop in-house for carrying out this kind of secondary analysis of the open access data sets. I'll get to the schema now. So for the ImmPort data model, it's like, how typically a database, relational database, we start with a study, and then we have the arms, which is like a control arm, treatment arm, the experiments that's getting carried out on the studies, and then the ontology. So this is just briefly showing you an idea about like data model for ImmPort, how we capture the data, and behind this is just a simplified model, but this is like, there are 100 tables, just the complexity that we heard this morning, and I will just reiterate that these tables, when you have 100 tables to go for, I mean, you're building the queries becomes pretty complex. Now I'll move on more into the focus areas and the data types in ImmPort. So we have about 413 studies with about 58,000 subjects, 62 diseases, data sets on those diseases, and we have tons of lab tests, and then if we break down into studies, as you can see here, we have a lot of allergy data sets, and then immune response, infection response, transplantation. We have a big amount of transplantation trials data, and these are all open access. And then on the experiment side, we support multimodal data. So we have a big number of flow cytometry data measuring the immune cell phenotypes and gene expression and other immune measurements. So we just support all different types of data which kind of facilitates the systems biology or systems level analysis of such data sets. And then total we have about over 46,000 total human subjects, slightly more females than males in this database. And then if you see the distribution of race, and this is universal, that we get a lot more data on Caucasians compared to the other races. But that's how it is, and right now there's a lot of initiative going on to focusing on the underrepresented communities as well. In terms of number for immunology clinical research studies, we have 291 and 122 clinical trials that's getting shared, and this is as of we share, the database gets updated quarterly, and we just updated in January 2020. So these are the numbers from there. So this is just to give you an idea about the studies, and there's a lot of data on flu vaccines, and then transplantation, dermatitis, atopic dermatitis, and also on healthy individuals. Just to give you a flavor that what we can do from these data sets, this is just an example of, as you can see here, this is a bubble graph where we have data from healthy subjects. The number is the number of participants, and then we have different cohorts like lupus cohorts, and then the different diseases here, aging, and then twin studies, and others, and microbiome, pregnancy cohorts. So what the common theme here is all of these subjects are vaccinated, but these are different cohorts, and the disease cohorts and the healthy cohort. Now, just to remind you, this data is coming from diverse sources, right? This is like all these investigators submitting the data, but no one is having a holistic view of these kind of data sets. So there's a lot of opportunities there to kind of evaluate it, and a little bit later I'm going to be giving you some of the examples of what we have been doing. So what is the purpose? So purpose, and where I come in here, is that I am a bioinformatician by training / data scientist, and we do the reanalysis of the clinical trials. So here is just a schematic or just a cartoon showing that there are primary investigators. They submit the data to the public repository, and then data scientists, they come in and they look at the data, and they combine the data and do meta-analysis, and then they got a novel insight depending on the question they're asking, and then they do some validations of the findings from the primary investigators, and then the new findings and then gets published, and that's called the secondary analysis or the meta-analysis. So next few slides are all about the repurposing projects that we had been doing in our group. So the first paper we got working on was about reanalyzing the clinical trial data. This was back in 2014, where we were this trial on ANCA-associated vasculitis trial was shared through ImmPort, and so we reanalyzed the data. So this trial was about, it was a randomized, double-blinded phase 2/3 trial comparing the rituximab to cyclophosphamide. These are the two drugs for treatments for induction of remission in vasculitis patients. And we have about 197 participants. And this paper was published in New England Journal of Medicine, where they reported that 63 of the 99 patients in the rituximab arm reached the primary endpoint, and 52 of 98 were about 53% in the cyclophosphamide group. So there were two treatment groups, cyclophosphamide group and the rituximab group, and this percentage is about the complete remission. So when we read the paper, we saw that some of the data got shared through ImmPort, so that was good. And then we read that 35 of the patients treated with rituximab and 47 of those who were treated with cyclophosphamide failed to achieve remission. So then the question that we asked is, in retrospective, can you measure any factors which might be predicting the response to therapies? So I'll just walk you through how we do this kind of secondary analysis. So we look at what data is available, and we saw that we have the primary investigators have shared the clinical data, and then some of the mechanistic data, the flow cytometry, and the ANCA-streaming titers. So we have flow cytometry data and the titers, so that kind of motivated us to go forward. But when you are doing secondary analysis, always I want to remind you that first step would be validating the results that have been published. So we did the validation step. So whatever the primary investigators reported, based on the data that was shared, we kind of were trying to recapitulate what they found. That's the very first step. And then, now coming to our questions, what you were interested in, we were trying to see if we can do some baseline analysis, meaning that these patients were recruited in the clinical trials and they were randomized. So they were either in the cyclophosphamide group or they were in the rituximab group. But no one did a cellular profiling of these patients at baseline before the treatment. So since we have the data available, then we started looking at all these immune cell subsets, and then we focused on the granular site subsets, and we saw that there were differences between the failure and success in each of these treatment groups based on the granulocyte subsets. And so we saw that some of these granulocytes were hypogranulated or hypergranulated, and this data or information is coming from the flow cytometry, because then they share that data with us. And so then, based on these 187 subjects, we, based on these profiles that we built, we then separated them into groups of, we call it granulatory index. So hypogranulatory means low GI index, high GI index, and then we were looking at the rituximab group, cyclophosphamide group, and you can see here that only 30% were getting complete remission if they have a if they are hypogranulated. Whereas those who have hypogranulated and were treated with cyclophosphamide, there was about 67% complete remission, and just the vice versa. On the other side, if they're hypergranulated, they were doing better with the rituximab arm. So just to give you an idea that what we did from this was that based on these findings, then we just proposed a personalized treatment on the basis of the granulatory index, and that's what is showing up here, that the current protocol was just randomized, recruit them in either of these arms, but then if you profile them, and in a profile therapy, you profile them at baseline based on their immune cell subsets, and then you get a better remission rate in each of these groups. So essence of this is what kind of novel analysis you can do when the data is getting shared, and this can better inform for your next clinical trial design to keep this in mind. Then based on this paper, so we published this paper, we got motivated, and we embarked on another project, and this time we built a resource called 10,000 Immunomes, and the motivation comes from, like, in the field of genetics, there's 1,000 genomes, that's a reference to go to, but in the field of immunology, there is none such references available. So we were trying to build a large, diverse, clean reference data set for human immunology, came up with the interactive data visualization, and also disseminate the standardized data or harmonized data to the community. So we do it so that community can build custom control cohorts based on their specification, and then this data can be downloaded. So if you get a moment, 1000immunomes.org, if you go to that website, you'll be able to download all of this data. So again, the motivation was immunoassays have not been reproducibly characterized for a sufficiently large and diverse healthy human code. So here the benefit is also diversity, since these are all crowdsourced data. So we published this paper in Cell Reports. Just to give you a little bit more information, we manually curated and subsetted the data. So typically when the data is getting shared, either you have a control arm, healthy arm, both arms. So we just subset on the healthy because we were focusing on the healthy subjects. And so we started with 85 studies within ImmPort, which comprised of 10,000 subjects. And then we had the site-up data, flow cytometry data, if you're familiar with these measurement techniques, and then cytokine profiling, gene expression data, we standardize the data, and then dissemination. Here is just the breakdowns of by age. So we do have a pretty big range of age here, and then distribution is by race, gender, and age. And these are different types of measurements, the number of subjects. Again, this is all the resources that we did from open access studies. Here is the website which you can go, 10,000 Immunomes. And so now I will show you one of the initiatives we did back in 2015 in terms of knowledge phase. And this is about the immune modulators in disease, integrating knowledge from the biomedical literature and gene expression. So we published this paper in 2016. I'll walk you through the steps that we did. So we extracted the PubMed records based on the mesh descriptors for disease, cytokines, cell types, and then we computed the mesh descriptors co-occurrence between the disease cytokine, cell types, and then we clustered the data. And on the site, we also got the gene expression. So GU is a database for downloading the microarray, open access microarray data sets. So we were looking at the disease-specific cytokine gene expression levels, and then we did the correlation analysis for this. Yeah, and so we extracted here the numbers. So we started with all the PubMed records, filtered based on human, and then the records with disease, cytokine, immune, is all based on control vocabulary like that, the mesh descriptors. And we were also looking at the role of like therapeutics and diagnostics. So and then therapeutics and diagnostic related records, and again cytokine cells and diseases. These were the entities we were interested in, and this is all about co-occurrences. So more we can say that this is a lexical semantics where you are only looking at the co-occurrences of these three entities of cell, cytokines, and disease. I'll show you some results here. So this is a knowledge model. So from the cytokines, we did the text mining from this PubMed therapy, and we were asking the questions about what is the role of the cytokine as a therapeutic target or a diagnostic marker, and the clinical data and expression data we got from these databases, and we were trying to integrate. And then we also looked at these interactions with co-occurrences for cell type, disease, cytokines, age, ethnicity, gender, diagnostics and therapeutics. And clinical data is like blood tests and flow cytometry, ImmPort, expression, curated knowledge. So basically we extracted all this information using this NLP technology. And so what we did? So based on, after we curating all this information, we built some networks and disease-centric networks interest of time, I'm just showing you some of these efforts that we did. So just a little bit deeper when we get into the clustering, I'm just showing you an example that based on this co-occurrence analysis, we were able to cluster the diseases based on the co-occurrence with IL-6 and TNF-alpha. So these are on your y-axis is number of occurrences of this relationship, and these are all the cytokines on the x-axis, and then cluster based on the diseases. There's another one where we were able to pull out food allergy, asthma, hypersensitivity reaction, type 1 disease, and the co-occurrence would be the cytokines. And again, a little bit more, we looked at all these group of diseases associated with interferon gamma and TNF-alpha. So we also asked about the diagnostics and therapeutics. So what's the role of the cytokines in diagnostics and therapeutics, and we looked at all these relationships. So this gives you an idea about like how much data mining you could do using the kind of knowledge base you can build, and this was done back in 2016 based on an NLP approach. Now I'll move in and spend a few minutes on Grakn, the vaccine knowledge base. And so for this, I just want to bring up again the slide that I had shown you earlier about the influenza vaccine studies in ImmPort, and that was my motivation for building this Grakn, the knowledge base. And so just to reiterate that these are coming from diverse sources, and there's a lot of value in these kind of the cohorts where you, with the common theme of vaccination or flu vaccine, they were all vaccinated subjects. So what are the challenges? So when I was trying to put together and trying to get a holistic view of all these cohorts, how can I do the queries? The challenges I had is these are crowdsourced, data was not standardized. Data is siloed, right? Because I'm not able to combine. This bubble graph looks pretty, showing you number of subjects from each cohort, but when I run the queries, it's pretty challenging. Interoperability, so that's another challenge, that the repositories are federated, meaning you have a gene expression data in one repository, and then you have the other repository for gene expression, and how you crosstalk between the databases. So that's another challenge, lack of data standards, which is challenging when you're trying to integrate the data. So for the better understanding of vaccination response in the realm of what I was showing earlier, I'm trying to understand what's the effect of age, gender, race on vaccination. There are a lot of papers who have been reported the effects of age, gender, and race, but then I would like to know more about immune response in vaccinated individuals with disease conditions like acute and chronic and so on, effect of medications on vaccination response. So that cohort was an excellent cohort for me, or group of cohorts that can address these questions, but then I need a tool which will help me to ask these kind of questions. And at the end, the overarching goal is to improve the standard of care. So I know that this group doesn't need introduction on knowledge graphs, and this morning we had a wonderful talk explaining the knowledge graph, connecting the dots. So as you can see here that, when you travel through the dots, basically what you're looking at is like what is the common relationship, and we all are familiar with this example where when we were growing up with the Star Trek and Star Wars, and what you are pulling up is like these are all, if you're interested in science fiction and interested to know which are the movies under this category. So basically knowledge graphs connect the dots, and you can travel through these dots to and run your queries. So basically knowledge graph limits the complexity and organize the information into data and knowledge, compose the facts and figures that can be used to provide contextual meaning searches. And so the way I started with that bubble graph and now moving into Grakn and trying to build a Grakn knowledge base, Here's the subject. Subject has attributes like age, gender, race, zygosity, because I had a twin data, and they said, yeah, there are genetic components which kind of contribute to the vaccine response. So we have monozygotes and dizygotic twins that influences the vaccine response. We have biosamples, and then this subject receives an intervention, either prevention, which is like the vaccine. Therapeutic means they are taking some kind of medications if they're in the disease group, and healthy as well. So basically a subject is a patient, and so then that's a clinical phenotype. And then on the vaccine side, I am kind of trying to put together the vaccine type, year, strain, and outcome. And outcome in this space is the antibody titers that you get after the subjects are vaccinated. So under biosample, we get a lot of data on the molecular, the DNA, or sequencing data, mRNA, protein, cells, and then microbiome. And just to give you an example of the health, there are different health conditions. For example, vaccinated pregnant women, or on the disease side, the categories are cardiac, autoimmune, asymptomatic, acute, chronic infections. So how are we all putting all this together in the schema, and then we start querying the data. So I am right now at a stage where I'm trying to build the Grakn knowledge base model, what I described earlier. So I started building this, the schema here, where the subject starting, and then the attributes, the biosamples that's getting collected from these subjects, and then biosample, for example, is a whole blood, and then when it goes to the experiment, maybe from the whole blood, we are isolating the serums or plasma. So pretty detailed, kind of a schema on how all this data is getting, like how we can structure all this data. And then on the vaccine side, all the information about the vaccines, and then the disease and health, and the clinical phenotype. This all data exists in ImmPort, but not in this schema. So I'm trying to get, because ImmPort is a structural database, so I run SQLs and extract the data, but then I need to standardize it, and then I will be able to simplify the queries across and ask the questions like, in the disease individuals and vaccinated, what kind of a response you say in across age, race, gender, and many such questions can be asked. So the take-home message is that you can take a holistic approach to analyzing clinical trials by combining multiple trials. Open access clinical trials are a valuable resource to evaluate new hypotheses, gain insights, inform a better trial design. I just described you 10,000 Immunomes, which is a reference data set generated by integrating individual data from the publicly available immunology studies. For example, if you don't have control in your experiment, you can go to these kind of resources, download the data, and can plot it against your own control or your experimental arm. And so these resources comes in handy. Knowledge graphs can be used as a semantic search engine, sparking new ideas. So I would like to leave you here, because for me, Grakn is just the beginning, and in coming months, I will start querying the data. So embrace open access clinical trials. Hopefully I was able to convince you, and I'll end with acknowledgements. ImmPort is, we work under Dr. Atul Butte. He is the director of in Baker Computational Health Sciences at UCSF, and he is also the lead, overall lead for ImmPort project. And Northrop Grumman is the company, those who help us curate all the data in ImmPort. I worked closely for the Grakn with Thomas Sabat, Sharu Safari, and Daniel really helped me in setting up my travel here. So definitely the ImmPort funding support comes from NIAID. And I would like to also thank the data providers. Without them, we won't be able to do the secondary analysis or get insight to these trials. And then the knowledge base that I showed, I worked with Nofar Giffman, who was back then a postdoc working in our lab. and right now she's in University of Manchester. So just wanted to show you that we started this work back in 2015 where we were building knowledge base, and now we have these advanced tools like Grakn, which we can leverage to ask a more simplify these queries for the question of interest. So with that, I would let's end, and happy to take questions.